Welcome to week 3, where we're going to talk about the so-called central dogma of biology and how viruses have adapted to circumvent that central dogma. Let's get to it. Hello everyone, Dr. G here again. You might be able to tell from the title of this video that we're going to chat a little bit about some basic biology today. Well, you'd be correct. However, while we will be discussing some of the terms that you learned in high school biology, which I hope are still familiar, we won't be going into a great deal of biochemical detail. Instead, we're going to focus on process, on mechanisms. More specifically, we're going to talk about the portion of the biological central dogma that applies to the COVID-19 pandemic. In fact, once we review the central dogma, we will talk about how the SARS-CoV-2 virus does not adhere to the central dogma. That said, we need to review some basics so that we can have a quality discussion about the virus that causes COVID-19. So, before we get started, will this be a general review of basic biological processes? Yes. Will this be an in-depth discussion of how those processes occur on the cellular and molecular level? Not exactly. We'll touch on some key points at that level, but we'll save most of that for another course. But for now, let's get started. To begin, you might be wondering what that term dogma means. Well, while it's used in many contexts, the Merriam-Webster dictionary defines it as something held as an established opinion. So, while the dictionary defines it as an opinion, there is a fair amount of evidence to make the central dogma of biology more of a tenet or a doctrine. You'll notice that all of these words fall short of asserting permanence or indicating that this is absolute. That's not an accident. Biology, like all science, is subject to change when we get more information. What I'm about to explain to you was basically considered absolute at one time, but then, like good scientists do, we study, we learn, and we modify as needed. All of that said, it's still important to know that the following explanation is mostly rule with a few exceptions. SARS-CoV-2 just happens to be one of those exceptions. But in order to understand the exception, you must first understand the rule. From a 10,000-foot view, the central dogma of biology can be described like this: DNA to RNA to protein. Yep, that's it. All this lead up for that, right? Okay, maybe that's not it. Well, let's zoom in a little bit, because while a 10,000-foot view or a 10,000-foot understanding might be sufficient for a Trivial Pursuit game, it's not necessarily the same as understanding. Since you are old enough to remember, you'll probably recall the statement, DNA is the blueprint of life. Well, that statement is correct. As you probably know, DNA stands for deoxyribonucleic acid, and it's made up of 4 nitrogenous bases oriented across a double-stranded sugar phosphate backbone. Did I just lose you? Yeah, I thought so. So here's the bit I was talking about in my introduction. My goal with this lesson is not to reteach the structure of DNA and RNA and proteins to you. My goal here is to help you apply what you're already familiar with. And if you'd like to talk about the 5' to 3' anti-parallel orientation of DNA, I'm happy to do that with you, but I'm more interested in keeping you, well, interested. Specifically interested in how the central dogma of biology applies, or doesn't apply, to SARS-CoV-2. Let's continue. As we said, DNA is the genetic blueprint of life. Having said that, the vast majority of the functions in a living organism are carried out by, or facilitated by, proteins. That's where RNA comes in. Well, messenger RNA to be precise. Messenger, or mRNA, uses the DNA blueprint to synthesize functional proteins. So remember the basic dogma that I explained, DNA to RNA to protein? That is the 10,000-foot view, but we need to zoom in a little bit further. In plant and animal cells, DNA is enclosed in a cell's nucleus. As cells divide, they copy DNA over and over as they divide. Cells, however, are also protein manufacturing plants. And while they're busy copying and dividing, cells are also synthesizing proteins. These proteins might be receptors that are designed to move chemicals in and out of the cell. They might be proteins that help cells communicate with one another. They might be receptors that allow material to bind to the cell in a very specific way. Cells will in fact make the proteins that they need to replicate themselves. The list of possible proteins and what they do is very long and really cool. Now DNA, however, doesn't have a way to make protein without help. That's where mRNA comes in. With the help of special structures, which are proteins by the way, and enzymes, which again are proteins, a very specific section of DNA called a gene is transcribed into a very specific form of mRNA. This process is known as transcription, occurs inside the cell's nucleus. The very specific mRNA that is created during transcription is going to leave the cell's nucleus and move into the cell's cytoplasm. Once it's in the cytoplasm, this specific mRNA holds the very specific instructions to make a very specific protein. This process is called translation. Translation is the process where mRNA is translated into a string of amino acids. That string of amino acids is called a polypeptide and eventually will fold up in a certain way to ultimately become a functional protein. In other words, the amino acids themselves and the order of those amino acids dictate how and what a protein looks like and how it functions. That very specific protein will be charged with a very specific job in the body. This is basically the central dogma of biology. So, what the heck does that have to do with COVID-19? Well, I'm glad you asked. Let's consider an example. In fact, let's consider an example called the ACE2 protein. What ACE2 stands for and what it does and how it does it is a little out of scope for this conversation, but it is important that you know that ACE2 plays an important role in helping people maintain a healthy blood pressure. It's also important that you know that ACE2 is an abundant receptor protein in our bodies and can be found on the surface of cells in our lungs and in our kidneys and our circulatory system and more. So, with that specific example in mind, let's apply it to our central dogma 10,000-foot view. So, we have a specific DNA gene sequence for ACE2, okay? That specific DNA gene sequence of ACE2 is transcribed by RNA polymerases into mRNA, which is then translated into polypeptides, which ultimately become the ACE2 receptor protein. Let's dive a little bit deeper into how this process occurs. And remember, we're only diving as deep as we need to. Earlier I mentioned the word enzyme. Although you've probably heard that word before, you may not really know what it means. An enzyme, which again is a protein, is a sort of biological catalyst. In other words, enzymes help to speed up a biological process or a reaction. Whenever you see the letters ASE or ACE at the end of a word in biology or in biochemistry, this is signifying that this object, this molecule, this thing is an enzyme, okay, this protein. Let's look at a couple of examples. Earlier we talked about cells dividing and DNA being copied over and over, right? Well, there's an enzyme that speeds up the replication of DNA, and that enzyme is called DNA polymerase. If you know a couple of glossary terms and break the word up, you can see that it basically defines itself. So you know what DNA is, right? Again, deoxyribonucleic acid. All right, hang on to that. A polymer could be described as a repeated sequence or a repeated series of monomers. So one unit of something is a monomer. If you have many monomers, then you have a polymer. Okay? And then finally we have the ASE suffix, which signifies it as a biological catalyst, or as we learned, an enzyme. So DNA polymer catalyst, or better, DNA polymer enzyme, or flip it around a little bit, DNA polymerase is an enzyme that polymerizes DNA. Back to our central dogma. In the following description, I've oversimplified the process and left out a few details, but in principle this process is simple, but as I've said, the biochemistry is complex and outside the scope of our conversation. But with that out of the way, let me describe the central dogma of biology as it's applied to the ACE2 receptor. DNA, which holds the genetic blueprint for ACE2, is replicated by DNA polymerase. The ACE2 gene, which is the DNA blueprint of ACE2, is transcribed into mRNA. So if mRNA is a polymer like DNA, what protein would you expect, what protein would you predict facilitates mRNA transcription? Yep, we already talked about that, RNA polymerase. An enzyme that polymerizes RNA, mRNA to be precise. Finally, the mRNA carries DNA's, we'll call it message, via mRNA out of the nucleus of the cell into the cytoplasm, where it interacts with more biological machinery to make, ultimately, the ACE2 protein. Now, it is not an accident that I used ACE2 as an example. This is not the last that you will hear of the ACE2 protein. But in order to find out why, you're going to need to keep watching. If you have questions, please don't hesitate to reach out to me. I know this is a lot in a short amount of time in this seminar course, but if you have questions, let me know. We'll see you soon.